Hey guys, welcome to chapter 13. This chapter is broken into two topics, topic 1 and topic 2. In topic 1, we're going to focus on glycolysis, and in topic 2, we're going to talk about the citric acid cycle. Chapter 14, we'll talk about the electron transport chain as well as photosynthesis. So that's where we'll finish up the process that you've learned before, as cellular respiration. But for this point, we're just going to talk about glycolysis in topic 1. So as I said, we're going to talk about the catabolism of sugar and fats, which includes the breakdown of food molecules, the whole molecules that come into your stomach as you ate lunch or breakfast or dinner even, and then we're going to walk through the steps of glycolysis. Here are your topic objectives. As always, if you have any questions, please let me know so we can talk about them in class or at another time if you want to set up office hour appointments. Usually you should, most of this should be fairly new to you, or most of this should be fairly familiar to you, but if you have any questions, please don't hesitate to ask. So as I said, we've talked a lot about processing energy all semester, and so what we're going to talk about in this section is how cells actually get this energy. We all will talk about how they have to have the energy to do all the reactions, but we need to talk about how they got this. And you can see from this pathway here, we go from the breakdown of food molecules through glycolysis, substrate level phosphorylation, to acetyl-CoA transformation, and finish with the citric acid cycle for this catabolism process. And then we'll talk about energy harvesting with the electron transport chain in chapter 14. So we have to start by breaking the food down, right? Every time we talk about cellular respiration, we talk about the glucose just being this perfect molecule that's ready to enter the system. Well, that's not always the case, right? That's not how we eat it. We don't just ingest straight glucose. We ingest all three types of macromolecules. We ingest three of the four types of macromolecules: proteins, polysaccharides, and fats. And all of these have to be broken down in the cells and in the gut before they can be brought into the cell. And then they'll enter into our energy harvesting in different areas, depending on what molecules they are and how ready they are to enter the process. And this is really important because if you remember in chapter 3, we talked a lot about how a lot of breakdowns and things like that can't happen in one step because of the massive amounts of energy demands. And so that's why it's important that we understand that there's several stages to breaking it down. So we start with stage 1 where we break down those large macromolecules into the subunits. Then we have stage 2 where we do the glycolysis process, and then stage 3, which is where we complete the citric acid cycle and the electron transport chain. Now remember, glucose will enter our cells as well as other macromolecules will enter our cells as through symports and other transporters that we've talked about. Remember the example we had in chapter 12 is the sodium glucose symport. So think about that as you're thinking about this process here. So let's talk about glycolysis now. Glycolysis, as you should know, literally means sugar splitting. So in this process, we're going to take one molecule of glucose, we're going to split it into two molecules of pyruvate. The goal of this process is to generate two NADHs, which is going to shuttle electrons to the electron transport chain. There's two phases. There's the investment phase and the payoff phase, which give us a net of 2 ATP generated, which is not a whole lot of energy for the cell. So remember, ATP generation is not the goal of glycolysis. The generation of the two pyruvates and the two NADHs are the goal of glycolysis. This occurs in the cytosol, and all cell types will do this process. Now we're going to walk through each of the steps here in the next several slides. And I'm going to point out to you the important aspects of them, so I want you to be familiar with it. While I don't expect you to memorize the intermediates and the structures, I do want you to understand the importance of each of the steps in this process. So the first thing we do is we need to get glucose into the cell. And once we have it in the cell, what's important? We want to make sure it doesn't go back out of the cell. And what have we learned is important about that? Well, we know that charged molecules can't cross the membrane very easily. So what happens here is the cell will use hexokinase and ATP to put a charged phosphate group onto the molecule of glucose, making glucose 6-phosphate. This essentially sticks the glucose and makes sure that it stays inside the cell so it can be further broken down. Otherwise, it could be transported back out. And it also generates an ADP, and a proton out of this process. The next step of our investment phase is when we go ahead and we're going to rearrange the molecule a little bit. We're going to go from a 6-carbon sugar in this ring form that you see here to it's still going to have 6 carbons, but it's going to be in a 5 carbon ring form. And you can see how this is being reshaped a little bit. And the purpose of this is that it gives us a sort of a mirror image of the ring on either side. And that's going to make it so it's easier to split later down the road. So it's just kind of a rearrangement step at this point in the investment phase. Now we're going to continue through our investment phase, and this is where we lose our other ATP. And that what we're going to do is we're going to put a mirror phosphate group on the other side of the molecule. Once again, with our goal being to be able to easily split this molecule down the middle and create 2 identical molecules. So this is the other place where we use that ATP. Any sugar that's going to be broken down has to pass through phosphofructokinase in order to proceed through this process. So it's really important. This step 3 is the gatekeeper of glycolysis. So in step 4, we're going to enter into sugar splitting, and this is where the sugar is actually split. Now the molecule is not exactly a complete mirror. As you can see in the ring form, there's 2 hydroxyl groups that are upside down from each other, which means that they're not exactly mirror images when the sugar is broken. What that does is it produces 2 different molecules at the sugar splitting stage. We have the glyceraldehyde 3-phosphate, G3P, and we have the dihydroacetone phosphate. Now the G3P can go ahead and proceed on to step 6, but what's going to happen is that the other molecule has to be rearranged, and that's what we see here in step 5. In step 5, triose phosphate isomerase will rearrange the molecule to transform this into G3P. Remember, G3P has to be the format of the sugar in order to continue through this process of glycolysis. Anything else will have to be transformed into it first. So then we reach step 6, and this is where we enter the payoff phase. In step 6, we transition into the energy harvesting phase. Up until this point, we've only been putting items in. At this point, we're going to start being able to take out our NADHs, our ATPs. We're going to start seeing the energy and the efforts that we put into it here. The goal at the end of our process is to obtain 4 ATPs and 2 NADHs in this payoff phase, and that will leave us with a net gain of 2 ATPs and the NADH. Now remember, we're going to have this process occur twice because we have 2 G3P molecules at this point. So everything from step 6 on is going to occur twice, even though we're only going to talk about it happening once. So in this step, what we see here is the NADH is gaining an electron to become, or the NAD plus is going to gain an electron, and we're going to lose that phosphate group to make NADH. And so we end up with 1,3-bis-phosphoglycerate. So the new sugar has 2 phosphates, and NADH has come off and now has a charged electron that it can go to the electron transport chain to use. So then we see here in step 7 that there's a transfer of the phosphate group between 1,3-bis-phosphoglycerate to ADP, and this is going to create our first ATP coming out of the system. So this is the first place where you see that phosphate, that high energy phosphate, is going to come off and make ATP. And remember, anytime we're generating this ATP, it's through substrate level phosphorylation. So what we're left here is a high energy phosphate group. You see that guy down there in yellow on the bottom, and he's still on this 3-carbon molecule. And in this step, the remaining phosphate ester linkage is moved so that the carbons are ready, and makes this a little bit more of a high energy to prepare to lose that phosphate group. So we see here that we're going to remove water once again and create another double bond in this molecule, and this is once again, we're all moving towards high energy, which is going to want to kick that phosphate off. So then here in step 10, we can see where we lose the phosphate via substrate level phosphorylation once again to create ATP. This is going to leave us with pyruvate, ATP, and as you saw, the 2 NADHs that came off earlier. So this is the end of our glycolysis process. So remember that it's all a step-by-step process, with our main goal being to have 2 pyruvate molecules to enter the citric acid cycle and 2 NADH molecules to go to the electron transport chain. We generate a net of 2 ATP that will then be able to be used in other functions in the cell, but that ATP is just a side bonus. Remember, the goal of glycolysis is not to make ATP, but it is to create the pyruvate and the NADHs. So let's talk a little bit about substrate level phosphorylation. This is an example of this is an example of a coupled reaction. And we can see this highlighted here on this slide with steps 6 and 7 of the glycolysis. And you can see that the covalent bond is formed between the G3Ps and this sulfhydryl group of the cysteine side chain of the enzyme G3-phosphate dehydrogenase. And what this does is it helps us couple these reactions so that between steps 6 and 7, when we have these big changes in the molecules, we end up with a favorable reaction energy of -3 kilocal per mole. So it's really important to understand that that's how this substrate level phosphorylation is happening, is that we are coupling these reactions to create the ATP as well as drive forward the step from 6 to 7. So lastly, let's review glycolysis. As I keep saying, the net ATP production is 2, but remember that's not our goal. And it's net production because we have 2 invested and 4 produced for a total net of 2. We end up with 2 NADH molecules, and these are shuttled to the electron transport chain. Now, there's a couple important steps. As I said, I don't want you to memorize everything in this, but I want you to know what these steps 1, 3, 5, 6, and 7, because these are the important steps in glycolysis as far as getting ready for downstream events. So it's really important you understand that and review that coupled reaction slide with 6 and 7, and let me know if you want to talk about that a little bit more in class as well. Because it can be a little bit tricky with that substrate level phosphorylation. But those are the stages that you need to understand how they work, because these are setting up the groundwork for future events down the way. So this is the end of glycolysis. In the next topic, we're going to talk about the citric acid cycle. So as always, review your objectives and let me know if you have any questions about this part of chapter 13.